Welcome to the first in a series of videos in which we want to show you step by step what will happen to the insects you collected for the School Malaise Trap program. Once your package arrived here at the Biodiversity Institute of Ontario, your collecting bottles will be passed on to our collections group for sorting and imaging. The staff records details on the collection location from each bottle and compiles the weather data you recorded. Next, the contents of each bottle are poured into a sorting dish, and, using a microscope, every specimen is counted for each trap. Our staff then attempts to pick as many different species as possible and selects up to 285 animals for our DNA barcoding work in the lab. This process will eventually lead to about 18,000 individual insects for which we will try to obtain a DNA barcoding. For every species we analyze at our institute, we also make at least one photograph. This will help researchers later on, for example, if they want to check species identifications. All images are stored in the same database as the DNA barcodes. Our collections group has various equipment that helps them to photograph organisms of different sizes. Specimens collected for the School Malaise Trap program are either photographed with conventional SLR cameras, or in case they are smaller, we use a setup that contains a microscope that is connected to a digital camera. The microscope enables us to photograph animals that are often smaller than a millimeter. Once all these steps are completed, specimens will be moved into a DNA tissue place, but this we will show you in the next video. All the specimens that you collected will find a final resting place in our storage facilities. In this collection, we can house about 1 million insect specimens for a very long time. Every barcoded animal is stored in such a collection, for example in a museum or at our institute. Most of the insects will be pinned and sorted into a box, which together with a few other boxes will go into a drawer in one of the cabinets shown here. All the specimens that we can't put on a pin will be stored in similar cabinets. The animals will be stored in glass jars that are filled with ethanol. This is how we ensure that researchers can come back to a particular sample at any time in the future. Our next video will show you how we extract DNA from your samples. Thank you for watching.